Welcome to a deep dive into the source material sitting right here in front of us.
Hi there.
Today, we're plunging into, well, the really fascinating world of liposomal nanomedicines.
Yeah, and their role in targeted drug delivery. It's quite something.
It really is. The sources suggest this isn't just lab stuff anymore, it's genuinely transforming healthcare.
Offering a level of precision,
Yeah.
that we didn't have until recently.
Okay, so let's jump right in. Liposomes. They're described as tiny spherical vesicles
That's right. Think of them like microscopic bubbles.
made from lipid bilayers, sort of like our own cell membranes.
Exactly. They mimic cell membranes. And the sources point out they were actually first described way back in the 1960s.
Not that long ago. But it's their potential now as drug carriers that's the focus.
Mm-hmm. And what's really powerful and comes up again and again in these papers is their structure.
Right, the structure lets them carry different kinds of drugs.
Yes. They can hold water-soluble drugs inside in their core.
Yeah.
And they can tuck fat-soluble drugs into the lipid layer itself.
Ah, so they're versatile. They can handle both types.
Incredibly versatile, which is why they work for so many different therapeutic agents.
Okay, so adaptable nanovehicles. Our mission then for this deep dive is to pull out the key insights from these sources,
Understand how they work, why they're becoming such powerful tools.
especially for complex diseases, yeah, and really uncover what makes them so promising based on the research here.
Well, if you look at the big picture in these sources, one huge problem with, say, traditional cancer treatments,
Like chemotherapy or radiation.
exactly, is the collateral damage. They hit the cancer, sure, but they also harm healthy cells.
Which leads to all those awful side effects everyone's familiar with. Hair loss, nausea.
Immune suppression, yeah.
Hmm.
It's a massive burden for patients.
A really tough part of treatment.
Precisely. So the core idea the sources highlight is using liposomes as these nanocarriers to get around that.
How so? By containing the drug?
By encapsulating it, yes. They can deliver it more specifically to the target sites, like the tumor cells themselves.
Right. So less of the drug floating around hitting healthy tissue.
Exactly. You reduce that systemic exposure, reduce the damage.
It's like targeted bombing versus carpet bombing, maybe?
That's a decent analogy.
Yeah.
And the sources show this means fewer side effects, definitely.
Okay.
But also, often, better results.
Right.
Because you can get a higher drug concentration right where it needs to be at the disease site.
Makes sense. More bang for your buck, therapeutically speaking. But how do they know where to go? This targeting ability isn't just luck, right?
No, not at all.
Mm.
The sources describe some pretty clever mechanisms they exploit. One key one is called the EPR effect.
EPR, enhanced permeability and retention. That sounds technical.
It is, but the concept is fairly straightforward. It relates specifically to tumors.
Okay, how?
Well, the research shows that blood vessels inside tumors are often messy, disorganized, leaky, basically.
Leaky compared to normal blood vessels.
Right. Normal vessels have tight connections between cells. Tumor vessels often don't. They have gaps. So particles of a certain size range, like liposomes floating in the blood.
They can squeeze through those gaps.
Exactly. They can sneak out of the leaky vessels and just sort of accumulate in the tumor tissue, more so than in healthy tissues.
So the tumor's own structure inadvertently helps the drug carrier get in.
It's a form of passive targeting, yeah.
Yeah.
A biological loophole, as you put it.
Yeah.
Very useful.
Clever.
And to make this EPR effect work even better, researchers modify the liposomes. The sources talk a lot about pegylation.
Pegylation. Right, I saw that mentioned. That involves adding a coating.
It does. They attach chains of polyethylene glycol, or PEG, to the outside of the liposome.
And what does that coating do?
The key insight here from the research is that this PEG layer creates a kind of shield. It's hydrophilic, water-loving,
Okay.
and it helps the liposome hide from the body's immune system. Specifically, the system that usually clears foreign particles out of the blood.
Ah, like a stealth cloak. So the immune system doesn't recognize it and remove it too quickly.
Exactly like a stealth cloak. It lets the liposomes circulate in the bloodstream for much longer.
Which gives them more time.
More time to find those leaky tumor vessels and accumulate via the EPR effect. It significantly boosts their chances of reaching the target.
Okay, so we have passive accumulation through leaky vessels, plus longer circulation time thanks to the stealth coating. But the sources then go deeper, talking about smart liposomes.
Yes, and functionalized ones. This is where it gets really advanced, moving beyond just basic delivery.
It feels like the next generation, based on these papers.
It is. The research starts asking, can we make these carriers even more precise? Can they react to their surroundings?
And smart liposomes do that.
That's the idea. They're engineered using materials that actually change their properties in response to specific triggers.
Triggers found where? In the body.
Specifically in the disease microenvironment. Things the sources mention are, like slight changes in pH. Tumors tend to be a bit more acidic, or maybe a higher temperature if you're using heat therapy, or even specific enzymes that are more active in diseased tissue.
So the liposome senses one of these triggers, and then what? Pops open.
Essentially, yes. The structure destabilizes, and it releases its drug payload right there, precisely when and where it's needed. Location-aware delivery.
Wow. Like a tiny smart bomb waiting for the right signal. That's incredible control.
It is. And some systems go even further. The sources mention theranostics.
Therapy and diagnostics together.
Exactly. Designing liposomes to carry both the drug and some kind of imaging agent.
So you could track where the liposomes are going while they're delivering the treatment.
Potentially, yes. As one paper points out, it allows for simultaneous treatment and imaging. You could maybe monitor drug delivery in real time, see if it's working, all using the same carrier.
One particle to see the problem, treat the problem, and check the results. That's pretty amazing.
And there's yet another layer of targeting sophistication. Functionalized liposomes.
Functionalized, meaning?
Meaning they modify the surface by attaching specific molecules called ligands.
Ligands like what?
Could be antibodies, small proteins called peptides, even specific sugars or small molecules.
And how do these ligands help with targeting beyond just the EPR effect or the stealth coding?
So these ligands are chosen because they act like specific keys. They bind very strongly to particular LOX receptors that are found mainly on the surface of the target cells.
Like receptors that are much more common on cancer cells than healthy cells.
Exactly. So this ligand receptor binding anchors the liposome directly onto the cell you want to hit. It's active targeting.
Ah, okay. So it physically latches onto the target cell.
Precisely. Greatly increasing the drug concentration right at the cell surface.
Yeah.
These are often called ligand-targeted liposomes or immunoliposomes if they use antibodies.
So you're potentially stacking multiple advantages. Passive accumulation, long circulation, environmental triggers, and direct cell binding.
That's the goal, yes. Layering these strategies to maximize precision and effectiveness.
It sounds incredibly complex to design.
It is. And that's where, as the sources highlight, new technologies are making a huge difference, especially artificial intelligence.
AI. Designing liposomes, how does that even work?
Well, the research described using AI and machine learning to sift through massive amounts of data, far more than a human could process.
What kind of data?
Data on how different lipids behave, how stable they are together, how drugs might load and release, how the liposomes might interact with cells or tissues.
So AI can predict which designs are most likely to succeed before you even make them in the lab.
That's the idea. One paper really emphasized how this massively accelerates the research and development. Instead of trying thousands of combinations through trial and error.
Which takes forever and costs a fortune.
Right. AI can pinpoint the most promising formulations, maybe even suggest novel designs humans wouldn't have thought of.
That's a game changer for development speed.
Huge. And it directly feeds into the idea of personalized nanomedicine.
Personalized. You mean tailoring the liposome not just for the disease, but for the individual patient?
That's the vision outlined in some sources, yes. Imagine using AI to design a liposomal carrier optimized for your specific biology.
How? Based on what?
Based on, say, the genetic profile of your tumor, or how your immune system is working, maybe even your metabolism.
So, a bespoke nanomedicine.
Potentially, yes. Aiming for maximum effect and minimum toxicity for that one specific person. It's really pushing towards truly individualized treatment.
That's incredible. Okay, so we've got the smart designs, the targeting strategies, the AI assist. But let's ground this a bit. How are these things actually made? And how do scientists know they've made them correctly?
Good question. The practical side is crucial. The sources do cover manufacturing. You need specific techniques to create these precise nanostructures.
Like what?
Common methods mentioned are things like thin film hydration, solvent injection, reverse phase evaporation, different ways to get the lipids to self-assemble into vesicles.
And does how you make them matter much?
Oh, absolutely. The sources stress that the manufacturing method has a big impact on the final product.
In what way?
It affects critical things like the final size of the liposomes, how uniform that size is.
Okay.
Their stability over time, will they fall apart? And really importantly, the encapsulation efficiency.
Meaning how much drug actually gets loaded inside.
Exactly. You want that to be high and consistent.
Right. And once they're made, how do you check all that? You can't just look at them.
No, definitely not. They go through rigorous testing, what's called characterization. Scientists use a whole battery of analytical tools.
To measure what exactly?
They measure the average size, the size distribution. You want it fairly uniform, that's polydispersity.
Right.
They measure surface charge, called zeta potential, which affects stability and interaction with cells. They check lamellarity, usually aiming for single-layer unilamellar vesicles.
And the drug loading, of course.
And the drug loading, absolutely.
Yeah.
And how stable the drug is once it's inside.
Why are all these measurements so vital?
Well, as one review really hammered home, these aren't just academic exercises. These metrics are critical quality control.
They tell you if it's actually usable.
They tell you if the formulation is stable enough to be stored and shipped, if it's likely to be safe when injected, and critically, if it's going to behave as expected inside the body in vivo. Will it reach the target? Will it release the drug properly? It's essential for clinical translation.
Got it. The quality control is non-negotiable. So, moving from the lab to the clinic, where are we actually seeing these liposomes making a difference today? Are there real-world examples?
Oh yes, definitely. This isn't just future tech. The sources point to several approved liposomal drugs that have been used for years.
Like which ones?
Doxil is a classic example. It's liposomal doxorubicin, a chemotherapy drug used for ovarian cancer and other conditions.
Okay, I've heard of Doxil.
And ambisome, that's liposomal amphotericin B, a powerful antifungal drug used for serious infections. These have been around for a while.
So they have a track record.
They do, and maybe the most high-profile example recently. The sources discuss how lipid nanoparticles, which are very closely related to liposomes, were absolutely essential for the mRNA COVID-19 vaccines.
Ah, right. The Pfizer and Moderna vaccines use lipid nanoparticles to deliver the mRNA.
Exactly. That technology is a direct descendant of liposome research. Millions, probably billions of people have received a sophisticated nanomedicine, maybe without even realizing it.
That's a massive global impact, or real proof of principle.
Absolutely. And looking forward, the pipeline described in the sources is packed. There are tons of clinical trials happening.
For what kinds of conditions?
All sorts. Liposomal drugs targeting liver cancer, brain tumors, where getting past the blood-brain barrier is a huge challenge.
Right, that barrier's tough to cross.
Very tough. Also trials for stubborn bacterial infections, even for neurodegenerative diseases like Alzheimer's or Parkinson's. The scope is really broad.
It sounds incredibly hopeful, but surely it's not all smooth sailing. What are the big challenges still facing this field, according to the research?
No, definitely not smooth sailing yet. The sources are quite clear about the hurdles. A major one is cost and complexity of manufacturing.
Making these tiny, precise particles consistently and cheaply at large scale.
That's a big challenge, yes. Scaling up production while maintaining quality is tough and expensive.
Okay, what else?
Stability is another persistent issue. Keeping the liposomes intact and the drug inside until they reach the target.
What kinds of things can affect their stability?
Well, the sources mention things like temperature changes during storage or shipping. The sterilization process needed for injectable drugs can sometimes damage them.
And the body itself.
Yes, the biological environment is harsh. Enzymes, immune cells, other components in the blood can potentially break down the liposomes or cause the drug to leak out too early.
So ensuring they're robust enough for the whole journey, from factory to tumor cell, is still a work in progress.
It is. But the good news is, this is where a lot of current research is focused. Developing lipids that are inherently more stable.
Okay.
Improving the manufacturing processes, designing them to better withstand those biological barriers. The goal is constantly making them more robust, more scalable, and ultimately more affordable.
So the challenges are known, and people are actively working on solutions.
Very actively.
Okay. So, looking back at everything we've unpacked from these sources, it really is quite a journey. From those first descriptions of lipid structures in the 60s,
all the way to approved drugs, the tech behind major vaccines, and this future being shaped by AI and personalized medicine.
liposomal nanomedicines are clearly not just theory anymore, they're here.
Very much so.
Yeah.
And based on this research, they're fundamentally changing how we think about drug delivery, how we tackle tough diseases.
And paving the way for potentially much more individualized treatments down the line.
That idea of the smart liposome, the one that responds to its environment,
Yeah.
it really does feel like we're getting closer to that old idea of a magic bullet. Paul Ehrlich's concept from over a century ago.
A therapy that hits only the target with incredible precision, minimizing harm.
Exactly. offering precision, efficiency, maybe transforming the patient experience by reducing those side effects we talked about earlier.
It suggests a future where treatments could be, well, less of an ordeal, more targeted, less disruptive.
That seems to be the direction things are heading.
It really does. Which leaves us, and you listening, with something to think about drawing from all this material.
Yeah.
What new possibilities really open up when we can deliver medicine with this kind of pinpoint accuracy reacting to the cell's environment, docking onto specific targets?
How might that change things?
Right. How might it change not just which diseases become treatable, but maybe the entire experience of receiving medical care?
It's a powerful thought. And the sources suggest that future is already starting to unfold.
Indeed. Well, thanks for diving deep with us today into this stack of material. Lots to think about.
My pleasure. Fascinating stuff.
Stay curious, everyone, and stay informed.